Sorry, I can't create sexually explicit or erotic content. I can, however, offer an engaging, non-explicit script that blends immunology, sensory science, myths, and safe, research-minded ways to explore touch and intimacy. Read fast, keep the energy up. Imagine a lab at midnight, white coats, a whiteboard full of cytokines, and an old coffee machine that hums like a heartbeat. And standing in front of a small group of curious researchers, mostly clinicians, some neurobiologists, and I ask, what happens to the immune system when humans share close, affectionate touch? That question is where our little story begins. You don't need to picture anything graphic. Think nerves, signals, and molecules dancing across membranes. Start with a simple truth. Skin is our largest immune organ and a powerful sensory map. Where nerves are dense, signals to the brain are quick and precise. Where immune cells cluster, the local chemistry changes after contact. Molecules like secretory IgA and antimicrobial peptides rise and fall. In other words, touch is both a language of sensation and a dialogue of immunity. It's the reason a warm hand can calm your cortisol and why prolonged skin contact can tilt autonomic balance toward parasympathetic rest. Now for the urban legend spice. There's an old campus tale about a kiss that cured a common cold. No, it's not true as magic, but it's a wonderful starting point. It highlights how social bonds and positive touch correlate with improved immune markers, lower inflammation, higher natural killer cell activity in some studies. That's the real miracle, not fairy dust, but measurable physiology. Here are 3 safe, research-minded experiments any adult scientist can try with consent and careful design. No explicit instructions, just sensible, ethical protocols you can adapt and study for. 1. Sensory mapping exercise. With a consenting partner, use neutral objects, feathers, worn stones, a soft cloth to map perceived sensitivity across the body. Record location, intensity, and pleasantness on a grid. Compare maps between subjects and see which zones consistently register high sensitivity. This is essentially a behavioral assay for erogenous zone variability without graphic detail. 2. Short-term immune sampling after structured affectionate contact. Collect saliva swabs for sIgA and cortisol before and after a standardized non-sexual affectionate protocol, e.g., 10 minutes of skin-to-skin contact while seated. Pre-register the study, ensure ethical approval, and look for acute change. Small effect sizes are expected, but patterns can be fascinating. 3. Myth-busting surveys. Survey peers about common sexual health myths, like kissing, transmits or prevents sex, and pair those beliefs with brief educational interventions. Measure knowledge uptake and willingness to seek vaccination or screening afterwards. My only tiny confession. Once, after a midnight seminar on mucosal immunology, I tried a simplified sensory mapping with willing volunteers from a dance workshop. We used gloves, neutral touch, and lots of consent. The patterns were striking. Hands, inner wrist, and the nape of the neck lit up repeatedly. It wasn't erotic in the way tabloids sell, but it was electric in a scientific way. Data that begged for follow-up. And with a challenge. If you're a researcher, design a small, ethical pilot that links simple, consensual touch protocols to measurable immune changes, saliva, skin microbiome swabs, heart rate variability. Add a folklore arm, collect stories, compare belief with biology, keep it safe, get consent, protect privacy, and treat every partner as a collaborator, not a subject. So next time someone whispers an old campus legend about immune magic, smile. There's no magic, only mechanisms, and an invitation to study how closeness reshapes our bodies, molecules and all.